Hi, and welcome to Madness and More and the AP Biology series. Today, we're going to be looking at cellular respiration, which is part of the energetic units in AP Bio. Resources can be found in the description section. You can find a follow along notes packet, which will allow you to follow along with this lecture, and it serves as a notes template, which you can use for review. We also have a Kahoot available, which you can practice some questions with in case you need to review for a test. So let's begin. So here's just a few statistics and to review how the human body uses energy. The human body uses energy from ATP for all activities. Body maintenance alone uses 75% of all energy, and this includes our heart pumping, breathing, body temperature, digestion, active transport, which we looked at in the last video, which was chapter 5. And on average, 2,200 kilocalories are needed to supply enough energy for 24 hours. Mitochondria are 40% efficient at converting glucose to ATP, so this is where cellular respiration takes place. And a car engine is only effective at 25% at converting gasoline to kinetic energy. So the mitochondria are more efficient at converting resources into energy. A working muscle cell uses and regenerates about 10 million ATP every second. These statistics are not needed for the AP exam, but they're good to know and help you understand the importance of cellular respiration. Okay, so let's look at what cellular respiration is. Cellular respiration makes ATP and consumes oxygen. Most ATP is made when more oxygen is available. Oxidation, which is the breakdown of glucose into carbon dioxide and H2O, which is water, and cellular respiration breaks down glucose molecules and banks their energy in ATP. So breathing is a key aspect of cellular respiration because it supplies oxygen to cells and removes carbon dioxide. So carbon dioxide, as you may know, is a toxin to us, and it lowers pH, which means that it'll make our blood more acidic. So where is the energy in our food? We eat food for energy, but where is it found? It's found in the electrons, or covalent bonds, of organic compounds. Cellular respiration consists of happens when glucose loses hydrogen atoms as it is converted to carbon dioxide. Oxygen gains those hydrogen atoms as it is converted to H2O. The movement of electrons from one molecule to another is an oxidation reduction, which is called a redox reaction. Oxidation is a loss of electrons, while reduction is a gain. A good way to remember this is with the acronym OIL-RIG. The O in oil stands for oxidation, the I stands for is, and L stands for loss. Oxidation is loss. And for the reduction is gain, you can remember it as RIG. R stands for reduction, I stands for is, and G stands for gain. Reduction is a gain. So during cellular respiration, electrons are transferred to oxygen as the carbon-hydrogen bonds of glucose are broken and the hydrogen-oxygen bonds of water are made. Moving hydrogens represents moving electrons because hydrogen has one proton and one electron. As hydrogens are passed, their electrons lose potential, and that energy is put to work. As the elements are passed, they lose energy. So as the electrons you see here are passed on, they lose energy. And if a bond is broken, the bonding electrons can be used to build ATP. Use the energy from electrons falling from organic fuels down to oxygen. The key players in the oxidation of glucose are hydrogenase enzyme, which removes electrons in hydrogen atoms, and NAD+, which is an electron shuttle. Now, you may notice that most enzymes end in ase. So that is a good way to tell if something is an enzyme. I may have gone through this earlier, but whenever something ends with an ase, it is most likely an enzyme in biology. So NAD+ is a coenzyme made from niacin, which is B3 vitamin. The electrons transferred from glucose to NADH are then delivered to the electron transport chain. So let's look at the rest of the staircase analogy. Many people compare the electron transport chain to a staircase, and that is because the energy is going down, down. It bounces. So we'll go through the image after we go through the content. NADH passes electrons to an electron transport chain, which are a series of redox reactions to pass electrons from one carrier molecule to another. As electrons fall or get passed from carrier to carrier, and finally to oxygen, energy is released in small quantities. So here we see the amount of energy decreasing at every fall. So that's why this gives kind of a staircase feel to it, because it's going do, dee, do, dee, do. And oxygen is the final electron acceptor. There are 3 stages of cellular respiration. The first one is glycolysis, and it happens in the cytoplasm. It is where the breakdown of glucose happens, and glucose is broken down into pyruvate, which is half of glucose each, producing a small amount of ATP. This is a very hard topic to visualize, in my opinion, so I'm going to draw this out. So here we have glucose, so there is 6 of these. Glucose is made of 6 carbons, so I'm not going to write the C in here, just imagine them to be carbon. So all of these circles are carbon, and this whole thing is going to be glucose. Now, when I break this down into a half, so if I cut this up right here, so in the middle, that would mean that this half is a pyruvate and this half is a pyruvate. So that's why glycolysis results in 2 pyruvates from just 1 glucose, and 2 ATP are produced. after that, the prep for and the citric cycle or Krebs cycle happen. And these happen in the mitochondria. This completes the breakdown of glucose and produces a small amount of ATP, so it's 2 ATP. It also supplies the third stage of cellular respiration with electrons. So the Krebs cycle has to happen twice for each of the pyruvates. It happens for pyruvate 1, and then it happens for pyruvate 2. The third step is oxidative phosphorylation. It involves the electron transport chain that we saw on the past slide, and it uses energy released by falling or passing electrons to pump hydrogens across a membrane. It harnesses the energy of the hydrogen gradient through chemiosmosis, producing 34 ATP. There cannot be more than a total of 38 ATP. So this number, the 34 ATP, is not always accurate. It has the ability to produce 34 at max, but sometimes we have a leaky membrane, so you might have 30 ATP produced and not 34, or you might have 32. But in total, counting glycolysis, the Krebs cycle, and oxidative phosphorylation, you cannot have more than 38 ATP. So this is a quick overview of what cellular respiration looks like, the 3 steps. So first we see step 1 happening, where we have glycolysis happening, glucose going into pyruvates, and then you see that the pyruvate goes into your citric acid cycle. Citric acid cycle will happen twice, and then it'll go on to oxidative phosphorylation, which will give off 34 ATP. It should give off 34, but that's the max, and it'll result in a total of 38 max ATP. So in this image, we see the NADH electron carriers. FADH is also an electron carrier. Now let's look at the first phase of glycolysis in details. So glycolysis is basically sugar splitting. You do not need oxygen. This is the only step of cellular respiration, which is anaerobic, which means it does not need oxygen. So the glycolysis process harvests chemical energy by oxidizing glucose into pyruvate, and ATP is used to split one glucose into 2 molecules of pyruvate. 9 chemical steps happen: the reduction of 2 NADH pluses, which become 2 NADH, and the creation of 2 ATP through the substrate level phosphorylation. Substrate level phosphorylation is when an enzyme plucks substrate and uses it for ATP. So the first phase of glycolysis, there are 2 phases, includes the 2 ATP being used to energize glucose so that it can split. And then in the second phase of glycolysis, we have the steps 5 to 9, and these steps create 4 ATP, 2 NADHs, and 2 pyruvate. There is a net gain of 2 ATP because even though you created 4 ATP, we spent 2 ATP to split glucose into pyruvate. So that's why there's a net gain of 2 ATP. Now let's look at the prep for the citric acid cycle. Pyruvate is moved into the mitochondria and prepared for the citric acid cycle. Enzymes enzymes process pyruvate, releasing CO2, which is carbon dioxide, and producing NADH and acetyl-CoA. Acetyl-CoA is produced because two pyruvates went in, and it is a high-energy fuel for the citric acid cycle. And there are two acetyl-CoAs for every glucose produced. Now let's look at the Krebs cycle. So the first slide that we saw was just the prep, but this is the real thing. This is the citric acid cycle. So the citric acid cycle will generate 3 NADHs, 1 FADH, 1 ATP, and 2 carbon dioxide for each pyruvate. So because there are 2 pyruvates, this will be produced 2 times. The 2-carbon acetyl part of acetyl-CoA is oxidized, and the CoA part splits off and is recycled. 2C-acetyl, so the 2-carbon acetyl joins and it forms a 4-carbon oxaloacetate, and then there are redox reactions that take place. So citrate becomes a ketoglutarate because of the loss of carbon in carbon dioxide. And then after that, NADH plus becomes NADH. The A-ketoglutarate becomes succinate, which is because of the loss of carbon. And then again, NAD plus becomes NADH. And then 1 ATP is made using substrate level phosphorylation. And then after that, FADH and NADH are made. And then malate goes into oxaloacetate and goes to get another 2-acetyl group and starts the cycle over for the second pyruvate. So basically, oxaloacetate joins with CoA, acetyl-CoA, and then they make a 6-carbon chain. The 6th carbon is removed and creates a 5-carbon chain. And then the clipped off part of the another carbon is used to become 4 C. And then the joining with oxygen and 3 carbon are changed and then they are breathed out. So I know the second part that wasn't on the slides might have become a bit confusing, but the main things are what was on the slide. So oxidative phosphorylation, which is the electron transport chain, is where electrons from NADH and FADH travel down the electron transport chain using protein channels and electron carriers to oxygen, which picks up 2 electrons and 2 hydrogens to form water. Energy released by the redox reactions, which is used to pump hydrogen into the intermembrane space, creating a hydrogen gradient. In chemiosmosis, the hydrogen ions diffuse back through the inner membrane through ATP synthase channels, and ATP is made. Each molecule of glucose yields many molecules of ATP. Oxidative phosphorylation using electron transport and chemiosmosis makes up to 34 ATP molecules for each glucose molecule that enters cellular respiration. The max would be 38. And so 34 in oxidative phosphorylation, total, the max is 38. So the big picture is that without oxygen as the final electron acceptor in the ETC, chemiosmosis of ATP stops, and therefore no ATP is made and the cell dies. Alright, so this is a quick summary of cellular respiration. In my opinion, I think this unit is very hard because you need to remember all the details, and it actually seems intimidating at first, but when you study for it, it isn't that intimidating. The main things that you need to remember are what goes into each reaction and what comes out. So that would be, oh, how many ATPs go into that reaction, how many ATPs result, how are the electron carriers moving along through the process, and what comes out. So for example, in glycolysis, you have pyruvate coming out too. So just make sure that you know those things. Now, I recommend that you pause the video and you put in what step 1 is, step 2, and step 3. You put in which place they're located in the mitochondria for all 3 of them, and then also what they are. So what is the process? So you can pause the video, fill the chart in, and then I can review the chart. The chart can be found in a notes template in the description section if you want to print it. Alright, so the first step is glycolysis, and it takes place in the cytoplasm. And what is it? It is anaerobic. It oxidizes glucose. It generates 2 G3Ps first, which are changed into 2 pyruvates. And it makes 2 ATPs, and it makes NADH, which is carrier and carries electrons to step 3. Thing I did not mention in the chart yet was that 4 ATPs are used in glycolysis. 2 is a net gain. 2 ATPs are spent to make glucose into pyruvate. Now step 2 is the prep for and the citric acid cycle. It takes place in the mitochondria and it is aerobic. And 2 pyruvates are changed into acetyl-CoA. It makes carbon dioxide and makes NADH, and the 2 acetyl-CoAs enter the Krebs cycle and make carbon dioxide, NADH, and FADH. NADH and FADH are electron carriers. It results in 2 ATP. And the electrons are carried by NADH and FADH are the energy you need to run step 3. So all of the electron carriers are carrying electrons to step 3. Step 3 is oxidative phosphorylation. It takes place in the mitochondria, specifically the inner membrane. It is also aerobic, and it takes place with the electron transport chain. NADH and FADH drop off their electrons, and it happens through the passing of electrons, and the passing of electrons is usable energy to actively push hydrogen molecules out of the matrix. Each oxygen accepts 2 spent electrons and 2 hydrogens, and it's converted into water, and then chemiosmosis takes place, which is the diffusion of hydrogen molecules through ATP synthase, which allows a phosphate to be put into ADP, and that results in 34 ATP. So I hope that this summary was helpful. You can also use these summary tables as notes. You can add whatever other details that you need to add from the prior slides. This is just a quick summary. Now, there are many poisons that work by interrupting the critical events in cellular respiration. For example, rotenone, which is an insecticide, cyanide, and also CO, which is carbon monoxide, bind to electron carrier molecules and block the movement of electrons. Oligomycin, which is an antifungal, blocks the passage of hydrogen molecules through ATP synthase channels in bacteria, and uncouplers make the inner membrane leaky to hydrogen molecules, so no gradient is made. The end result of all poisons is that there is no ATP production, no energy is produced, and therefore the cell dies. Fermentation is an anaerobic respiration. So the first type of anaerobic respiration is lactic acid fermentation. Muscle cells and some bacteria perform this. NADH is oxidized back to NAD plus as pyruvate is reduced to lactate. In animals, lactate is sent to the liver and is converted back to pyruvate. In bacteria, lactate is used to turn milk such as yogurt or sour cream, turn soybeans into soy sauce, and cabbage into sauerkraut. So, you might actually feel lactic acid build up in your muscles. For example, if you run a long distance and you feel like a cramp, that is many times caused by lactic acid formation in your cells. And then the second type of of anaerobic respiration is fermentation. So alcohol fermentation happens when yeast are used for beer, wine, and bread making. Yeast cells oxidize NADH to NAD plus while converting pyruvate to carbon dioxide and ethanol. Carbon dioxide carbonates beer and champagne and also causes bread dough to rise. And when yeasts are confined to a wine vat, they die when ethanol levels reach 14%. Now, there is a syndrome called autobrewery syndrome, where the body ferments, uses alcohol fermentation for any foods consumed by the human that have gluten. So this results in high alcohol levels in the bloodstream of these of the people suffering with autobrewery syndrome. Autobrewery syndrome is rare though. Cells use many kinds of organic molecules as fuel. Carbohydrates, fats, and proteins are a few examples. They are converted to molecules that enter glycolysis or the citric acid cycle. Food molecules provide raw materials for biosynthesis. Cells use some food molecules and intermediates from from glycolysis and the citric acid cycle as raw materials to make organic polymers such as carbs, lipids, and also proteins. This process of biosynthesis consumes ATP. So where does the fuel or food for respiration come from? The answer is that it comes from photosynthesis. All organisms can harvest energy from organic molecules. Plants, but not animals, can also make these molecules from inorganic sources by the process of photosynthesis. The link between respiration and photosynthesis is that photosynthesis energizes cellular respiration, and cellular respiration makes photosynthesis possible. The process of photosynthesis is our next unit, so I hope that you're excited for it. Thank you for watching. Don't forget to check out our other resources in the description section. You can also fill out the Contact Us form or email me at mathesonmore@gmail.com to get in touch with any questions, any doubts for the series. Don't forget to explore the rest of our videos, which will help you prepare for the AP exam. Before we end this session, let's look through a quick exploration. How is a marathoner different than a sprinter? There are two types of human muscle fibers. There are slow fibers that can sustain repeated long contractions but can't generate quick power, and they rely on aerobic respiration to generate ATP. Cells with lots of mitochondria and myoglobin to carry extra oxygen are part of slow fibers. Fast fibers can contract more quickly and powerfully, but fatigue easily. They rely on anaerobic respiration and not oxygen to generate ATP. These cells are thicker, contain less mitochondria, and produce lactate, which causes aches and fatigue. So, guess which one is for which? Well, slow fibers could be used for a marathoner, and fast fibers are used by a sprinter.